Good morning, everyone. Today we are going to study the topic counter current mechanism or counter current multiplication, which is done in the uh, loop of Henle, collecting duct, and the vasa recta. Vasa recta, we know that it is a network of capillary over the loop of Henle. So this will be the Bowman's capsule. This is PCT, then descending loop, then ascending loop, then DCT, and the collecting duct. And over this, the efferent arteriole will make the capillary network, and the capillary network which is present on the PCT and DCT is called as peritubular capillary network, and which is covering the loop of Henle, it is called as vasa recti. So we know that urine is formed in the nephron, and in mammals the urine is produced in concentrated form. So mammals as well as including human beings, they are producing the concentrated urine, and this concentrated urine is concentration of the urine is taking place in the loop of Henle and vasa recta. So in the two limbs of loop of Henle, filtrate flows in opposite direction. This is the descending direction and this is the ascending direction. So the filtrate, when it is passing through PCT, it is flowing in opposite directions in the two limbs. So this forms a counter current. In the same way, in the vasa recta, which will be over the loop of Henle, there will also be two limbs, and in them also the blood is flowing in two different direction in the two limbs of the vasa recta. So this is flowing in opposite direction. Like in the descending loop of Henle, the filtrate flows in this direction. So in the vasa recta, the blood will flow in this direction. And in the ascending loop of Henle, the filtrate flows in this direction. So in the vasa recta, the blood will flow in this direction. And these are this flow of the current or flow of the filtrate in two different limbs in two opposite directions makes the counter current. So in two limbs of loop of Henle, filtrate flows in opposite directions, and this forms the counter current. And so this system of flowing a liquid in two limbs in opposite directions is termed as counter current mechanism, and this counter current mechanism helps in maintaining the concentration of the urine produced in the kidneys. Now, osmolarity of interstitial fluid. So now if we see this part of the nephron is present in cortex region, then this part up to this broader part of the ascending limb of loop of Henle, this is present in the outer medulla, and then the remaining part, that is the thin part of the loop of Henle, or we can say the U part of the loop of Henle, is present in inner medulla. So now the fluid surrounding the nephron, which is present in the medulla region, this fluid is called as interstitial fluid. So osmolarity of the fluid in the medulla initially at this place is medulla as well as cortex is initially it is 300 milliosmoles per liter. MOSM is the milliosmoles. Milliosmoles means number of particles in per liter. So 300 milliosmoles are there in the beginning when the filtrate is passing into the loop of Henle. Then it goes on becoming more and more concentrated, and by the time the filtrate reaches to the peak of the loop of Henle, or you can say the inner medulla part of the loop of Henle, it becomes 1200. And then again it goes on becoming hypotonic. So you can see from 1200 it became 700, then 400, then 200, then 100, then 100, again like this. So this osmolarity is responsible for the movement of water molecules in the descending loop of Henle. From descending loop of Henle, water molecules move out into interstitial fluid, and by the time this reaches up to the inner part of medulla, or you can say the peak of the medulla region, the so much water is coming out from it that the osmolarity becomes 1200 milliosmoles, means it becomes most concentrated. And then when this fluid passes into this loop of Henle, NaCl is coming out from here, and osmolarity goes on becoming lesser and lesser, because this part of the loop of Henle is permeable to NaCl, sodium and chloride ions. So, the of the interstitial fluid, as I have told, in cortical region is about 300 milliosmoles and reaches up to 1200 milliosmoles at the tip of pyramid in medulla. Then this concentration gradient is essential. Now, why this concentration gradient is maintained? Because it is essential for the excretion of concentrated urine. Now, this osmolarity is maintained by the different phenomena. So, in the first phenomena, what happens? Active transport of sodium ions, and along with this, there will be co-transport of potassium ion, means from interstitial fluid, potassium will enter into it and sodium and chloride ions will move out from the thick part of the ascending loop of Henle into interstitial fluid. Then impermeability of thick and thin parts of ascending limb is there for the water, means water cannot pass through ascending limb. Then blood supply to medulla by vasa recta, means the vasa recta is here, that is removing the excess water and solutes, means excess water is going into the blood from this part and solutes are going from this into the blood. So it is being removed. So what happens? This sodium chloride is entering into the interstitial fluid and making this more hypotonic, and this water is being reabsorbed by the blood capillaries of vasa recta. So water from here is being given to blood capillaries to maintain the volume of the blood, and the salt is given from the ascending limb into the interstitial fluid. So this maintains the hypertonic concentration of the interstitial fluid, because salts are being given into the interstitial fluid, and by the time this filtrate reaches into the collecting duct, the urea is also being given into this interstitial fluid, which is passing back into the blood capillaries here. So this is also being maintained by the, so it is making the urine more and more concentrated. Now because more and more urine is concentrated here, so water is more absorbed into the blood capillaries from this part. So this is how the volume of the blood is maintained and the urine is being made concentrated. Then now we will be studying about the renin-angiotensin mechanism of control of urine volume regulation. Now urine volume regulation we have studied about control by an ADH hormone. So that hormone is released from posterior lobe of pituitary gland, and when the osmoreceptors in hypothalamus detect an increase in osmolarity of blood and interstitial fluid, then the ADH hormone is released. This ADH hormone is increasing the reabsorption of water in collecting tubules. Then in our syllabus, renin-angiotensin mechanism is most important, which we have not studied. So this mechanism is controlled by juxtaglomerular apparatus. So this picture which you have seen, this is the juxtaglomerular apparatus. So this is the glomerulus, this is the afferent arteriole, and this is the efferent arteriole, and this is PCT, proximal tubule, and this is the part of DCT. So the position of glomerulus is in such a way that the part of DCT is in close contact between two afferent and efferent arterioles. So this part of DCT, when we see in the longitudinal section, it looks like this. So this is the DCT, this is PCT, and these are afferent and efferent arterioles. So we are seeing the longitudinal section through this. So in this longitudinal section, we can see this is the inner lining of the cells of DCT, in which the cells of the DCT, which are in close contact with the afferent arteriole and efferent arteriole, these cells become modified and they have cilia extensions, and these cells are called as macula densa. Then the inner lining of afferent arteriole, you can see these are the cells, and these cells are granular cells, and these cells are also called as, so these cells are also called as JG cells or juxtaglomerular cells, and or the renin producing cells or Lassus cells. So this is macula densa, this is the JG cells, and these are producing the enzyme renin. Then this is efferent arteriole, and these are the nerves which are initiating them to release the enzyme, the renin enzyme. And this is the tissue between the two afferent and efferent arteriole and in between the distal convoluted tubule. So these are the cells. So these have a role in maintaining the blood pressure. So when the blood flows through afferent arteriole, the lot of liquid part of the blood filters here and the thick blood flows here. So this thick blood needs the pressure. So to maintain the blood pressure of the blood vessels, this angiotensin system is working. Now, juxtaglomerular apparatus is a specialized structure formed by distal convoluted tubule and the glomerular afferent arteriole at the junction of afferent and efferent arteriole. It is located near the vascular pole of the glomerulus, and its main function is to regulate blood pressure and the filtration rate of glomerulus via renin-angiotensin system. Then about macula densa. Macula densa is a chemoreceptor cells in wall of DCT at juxtaglomerular apparatus JGA of nephron, which respond to changes in solute concentration, that is the sodium levels. Means when the sodium levels are getting changed, they are responding in DCT, and oxygen level in blood passing the glomerulus. Means the blood which is passing in the glomerulus, their oxygen and sodium levels are making the response for JGA cells to respond. Then this information is conveyed to JG cells. Means when sodium levels and oxygen levels are changed, the information goes to JG cells. Then these JG cells, that is the juxtaglomerular cells, or you can say renin producing cells, which are in the inner lining of afferent arteriole near the DCT. So these adjust the amount of renin output. Means how much renin has to be secreted. It depends on the volume of or concentration of sodium and oxygen passing in the glomerulus. Then JG cell, that is the juxtaglomerular cells, are what? These are specialized smooth muscle cells, act as mechanoreceptors which stretch in response to increase in blood pressure of afferent arteriole. Means when the blood pressure in the arteriole increases, they are stretching. Then they also synthesize and secrete the enzyme renin, which serves to activate angiotensinogen into angiotensin 1. Means angiotensinogen is a enzyme which is produced in the liver, and it is in the inactive form, and this is converted into its active form in the presence of enzyme renin. Then renin secretion increase by fall in sodium ion. Then prostaglandins, sympathetic autonomic nervous system, and decrease in blood pressure in afferent arteriole. Means when the renin is secreted, when the fall is there in sodium ions, fall in the prostaglandins by the effect of sympathetic or an ANS, and by the decrease in blood pressure in afferent arteriole. Means when the blood pressure in afferent arteriole decreases, when it will decrease, when the blood will filter into the glomerulus. So this will cause the secretion of renin. So now we will be studying about renin-angiotensin system. So now controlled by juxtaglomerular apparatus JGA or renin-angiotensin mechanism. It operates via multi-hormonal system called renin-angiotensin aldosterone system or RAS. So what happens when there is a decrease in glomerular blood pressure or blood flow in afferent arteriole of glomerulus decreases, then what happens? JGA, that is the juxtaglomerular apparatus, becomes active and releases hormone renin into blood stream. This renin hormone converts plasma protein angiotensinogen to angiotensinogen 1. This is produced by liver and sent into the bloodstream. So this is present in the plasma and renin converts this angiotensinogen to angiotensin 1 and further to a peptide angiotensin 2. This is a vasoconstricting hormone and this is converted in the presence of ACE enzyme, which is produced by lungs. This is in the presence of angiotensin converter enzyme. ACE means angiotensin converter enzyme. So these conversions of angiotensinogen to angiotensin 1 and to angiotensin 2 takes place in the presence of enzyme ACE, which is produced by lungs, and this is a vasoconstricting hormone. So this reduces blood pressure by, first of all, constricting efferent arterioles to increase glomerular blood pressure. Means when the blood is coming through afferent arteriole and efferent arteriole becomes constricted, what will happen? The glomerulus and the blood pressure in glomerulus will increase. It is also stimulated by PCT. Stimulates PCT to reabsorb sodium chloride and water. So by the release of this angiotensin 2, PCT will reabsorb NaCl and water. And it also stimulates adrenal glands to release hormone aldosterone. So aldosterone will, the release of aldosterone will promote water and sodium chloride absorption by PCT. It also increases aldosterone secretion, feeling of thirst, salt appetite, means feeling to eat salty things, and ADH secretion. So all these things will be stimulated by the secretion of renin. And when the renin will be secreted, aldosterone will be secreted by the adrenal glands, and the efferent arterioles will be constricted to increase glomerular blood pressure. PCT will reabsorb more sodium chloride and water, and this adds to increase both blood pressure and blood volume. So this is how angiotensin system is functioning to increase the blood pressure in the body. Now your portion for final exams will be the living world, biological classification, plant kingdom, morphology of flowering plant, anatomy of flowering plant, then cell, the unit of life, biomolecules, cell cycle, then transport in plants, mineral in plants, this one, respiration, plant growth and development, digestion and absorption, the topics which I taught, breathing and exchange of gases, also the topics which I taught. This will not be there. Then excretory products and elimination, all the topics which I taught. Then these three will not be there in our syllabus. So with this, I am ending our today's session. Thank you.